All right, so get this. We're going to dive deep into genetics.
Oh, wow.
Yeah, starting with, those tiny building blocks of DNA,
Okay.
all the way to the intricate 3D structures of chromosomes.
Hell's complicated.
It is. It's going to be a wild ride.
I'm excited.
We've got some really cool sources for this one.
Oh, yeah.
We're talking journal articles, textbooks, even some 3D models to help us visualize this stuff.
3D models, that's cool. I like visuals.
Me too. It's going to be mind-blowing.
So where do we even begin with something this complex?
Well, I think it's best to start at the very beginning.
Okay, that makes sense.
What exactly are nucleic acids?
Yeah, that's a good starting point.
They're like the instruction manuals for life.
Instruction manuals, huh?
Yeah, but written in a special code that tells cells how to build and maintain themselves.
And that code is made up of just 4 letters, right?
Right. The nitrogenous bases adenine, thymine, guanine, and cytosine.
DNA, right?
Yep, for DNA. RNA swaps out thymine for uracil, though.
Oh, that's right.
But yeah, from just those 4 letters, you get the whole story of life.
It's kind of like a language, isn't it?
Yeah, it's like those 4 bases are the keys on a piano.
Okay, I can see that.
Depending on how you play them, you can get a simple lullaby or a complex symphony.
I like that analogy.
Now, to make those words in this genetic language, those bases are combined with a sugar and a phosphate to make nucleotides.
Nucleotides, those are the building blocks, right?
Exactly. They're like the Lego bricks of DNA and RNA.
So we're building with Legos now.
It's a good way to think about it.
I like it. So just those 4 bases and a couple of other things are all it takes to build everything from a bacterium to a blue whale.
It's amazing, right?
It is. It's mind-blowing.
And here's where it gets even more interesting. The type of sugar determines whether a nucleic acid is DNA or RNA.
Okay, tell me more about that.
DNA uses deoxyribose, while RNA uses ribose.
Ribose, those names sound familiar.
They're very similar.
What's the difference?
Just one oxygen atom. Seriously, just one oxygen atom makes that much of a difference.
Yep, that tiny change has huge implications for their roles.
Okay, I'm intrigued. How does that work?
Well, DNA with deoxyribose is super stable.
Stable, so that's good for long-term storage.
Exactly. Perfect for holding all that genetic information.
Okay, that makes sense.
RNA with ribose is more reactive.
Reactive, so it's more of a go-getter.
Yeah, it plays all sorts of roles in building proteins and transmitting genetic messages.
So like the busy worker bee of the cell.
Exactly. Now let's zoom in on DNA and that iconic double helix structure.
Okay. Yeah, that one's famous.
Watson and Crick unveiled this elegant structure back in 1953.
1953, that's a while ago.
I know, right? It was a huge discovery.
So remind me, what does it look like again?
Picture 2 strands twisting around each other like a spiral staircase.
Okay, I can see that.
The sugar-phosphate backbones make up the outer rails, and those base pairs, A with T and G with C, form the steps.
So like a twisted ladder.
Yeah, kinda.
Pretty simple, yet so important. And don't forget, the arrangement of those bases is what makes up the entire genetic code.
It's like a secret language.
It is. But there's another crucial detail here. Those 2 strands are anti-parallel.
Anti-parallel, what does that mean?
It means they run in opposite directions.
Oh, I see.
Like head to toe, which is super important for replication.
Why is that so important?
Well, each strand can act as a template to create a new matching strand.
Oh, that's clever. Like a built-in backup system.
Exactly, and that's how genetic information is passed on accurately during cell division.
Makes sense. But hold on, DNA isn't always this perfect staircase, is it?
You're right, it's more flexible than you might think.
Oh.
Yeah, it can actually morph into different shapes depending on the conditions.
Really? So it's not just a rigid ladder?
Nope, it can bend and twist. The BDNA form is the most common.
BDNA, is that the one we see in pictures?
Yeah, the classic double helix.
Okay.
But DNA can also form aDNA or zDNA.
Wow, so many different forms.
It all depends on things like humidity or what proteins are around.
So it's more like a dynamic, adaptable structure.
Exactly. And it can get even weirder. We're talking bends, loops, even triple helices.
Okay, now you've got me hooked. Tell me more about these weird shapes.
Well, imagine a string of adenine bases. Those can cause a bend in the helix.
A bend, like a kink in a hose.
Yeah, exactly like that.
Interesting. And what's the purpose of that?
Well, it affects how DNA interacts with proteins and how tightly it's packed.
So those kinks aren't just random, they're important.
Absolutely. Every little detail matters.
Wow.
Now, imagine a DNA sequence that's a palindrome.
A palindrome like race car.
Yep. These palindromic sequences can make the DNA fold back on itself.
Fold back on itself, so it's like mirroring itself.
Yeah, forming hairpin or cruciform structures.
And those structures do something?
They can regulate gene expression or even play a role in DNA replication.
Pretty cool how such a simple sequence can do so much.
And get this sometimes, DNA can form a triple helix.
Triple helix, so 3 strands instead of 2.
Yep. It involves a special type of base pairing called Hoogsteen base pairing.
That's new to me.
It's less common but still important, especially in processes like gene silencing and DNA repair.
Okay, I'm starting to see that DNA is much more complex than I initially thought.
Right. But here's a big question. How does all that DNA, which can be super long, fit inside a tiny cell?
Yeah, that's a good question. It's like stuffing a really long string into a tiny box.
It is. And the answer is DNA supercoiling.
Supercoiling. What's that?
Imagine taking a coiled phone cord and coiling it up even further.
Okay, so like a coil within a coil.
Exactly.
Right.
That's basically what happens to DNA to make it more compact.
Clever. It's like nature's ultimate space-saving hack.
It is, but it's not just about saving space.
Oh, there's more to it.
Supercoiling also affects how easily DNA can be accessed for things like replication and transcription.
Okay, that makes sense.
It's like organizing the files in a cabinet so you can find what you need quickly.
I can see that. And what controls this supercoiling?
Enzymes called topoisomerases.
Topoisomerases, what a mouthful.
I know, right?
So what do?
They're like molecular scissors and glue.
Scissors and glue. Interesting.
They cut and rejoin the DNA strands to adjust the level of supercoiling.
So they're like the master organizers, making sure everything is packed just right.
Exactly. Now, let's zoom out a bit and talk about how DNA is organized within cells.
Okay, sounds good.
Starting with simple cells, prokaryotes like bacteria.
Bacteria, those are the simple ones.
Right. They don't have a nucleus like our cells.
So where's their DNA?
It's all concentrated in a region called the nucleoid.
Nucleoid, so like a central DNA zone.
Yeah, but it's not a separate compartment like a nucleus.
Okay, so it's more like a designated area.
Right. But even though it's not as structured as a nucleus, bacterial DNA is still organized.
How so?
Thanks to these special proteins called nucleoid-associated proteins, or NS.
NFPs, what do?
They help bend, wrap, and organize the DNA, keeping it from getting tangled.
So they're like tiny librarians, keeping the DNA library in order.
I like that analogy. And there are different types of NAPs, each with a specific job.
Oh, like what?
Well, HU is a histone-like protein that makes DNA more flexible.
Flexible, so it can bend and twist easily.
Yeah, exactly.
Okay, what else?
There's HNS, which does the opposite. It stiffens DNA.
So it keeps things from getting too floppy.
Yep. And then there's IHF, which can introduce sharp bends in DNA.
Sharp bends, why would you want that?
It influences gene expression, which genes are turned on or off.
So it's a coordinated effort with each NFP playing its part.
Precisely.
Yeah.
Now let's shift gears and talk about eukaryotic cells, like ours.
Okay, so now we're getting more complex.
Right. These cells have a nucleus, and that's where we find chromosomes.
Chromosomes, those are the X-shaped things.
You got it.
Yeah.
And chromosomes are made up of chromatin.
Chromatin, remind me what that is again.
It's that complex of DNA and proteins.
Right, I remember.
The basic unit of chromatin is the nucleosome.
Nucleosomes, they always look like beads on a string in textbooks.
That's a great way to visualize it. Each bead is a core of histone proteins, and the string is the DNA wrapped around it.
So the DNA is neatly spooled around these histone cores.
Exactly. It's an incredible level of organization.
It is. But is all chromatin packed the same way?
Great question. There are different levels of packing, resulting in two main forms: euchromatin and heterochromatin.
Euchromatin and heterochromatin, I always get those mixed up.
Think of it this way. Euchromatin is loosely packed.
Loosely packed, so it's easy to access.
Exactly. That allows genes to be expressed.
Okay, that makes sense.
Heterochromatin, on the other hand, is tightly packed.
Tightly packed, so like a storage unit.
Yeah, it usually contains genes that are not actively being used.
So it's like having different levels of access to the genetic library.
Exactly. And this packaging can change depending on the cell's needs.
Wow, it's like a dynamic filing system.
It is. But there's another layer of organization we need to consider: chromosome territories.
Chromosome territories, what are those?
Each chromosome actually occupies a distinct region within the nucleus.
Wait, so they have their own designated spots?
Yep. It's not random at all.
That's critical. Why is that important?
Well, it helps regulate gene expression and prevents chromosomes from getting tangled.
So it's like each chromosome has its own workspace.
Exactly. But sometimes we can actually see gene expression in action.
Really? How is that possible?
There are these special types of chromosomes, Lamb-Brush chromosomes and Polytene chromosomes, where gene expression is visually spectacular.
Okay, now you're just teasing me. Tell me more about these special chromosomes.
Let's start with Lamb-Brush chromosomes.
Okay, where do we find these?
Imagine peering through a microscope and seeing these vibrant, feathery chromosomes. That's what Lamb-Brush chromosomes look like.
Feathery chromosomes, that sounds amazing. Where do they exist?
They're typically found in developing egg cells of amphibians and some other
The
organisms.
amoebians, interesting.
They have a central axis with loops of DNA extending outwards, giving them that feathery appearance.
Why do they have those loops?
Each loop represents a gene that is actively being transcribed.
So we're literally seeing genes in action.
Exactly. And the size and shape of those loops can tell us how active a gene is.
That's incredible. So it's a dynamic picture, not just a static image.
Exactly. It's like watching a gene in action.
Wow. Now tell me about those Polytene chromosomes.
Collagen chromosomes are giant chromosomes found in certain insect tissues.
Giant chromosomes, how big are we talking?
They can be hundreds of times larger than normal chromosomes.
Oh, that's big. Why are they so giant?
They're formed when DNA replicates multiple times without cell division.
So it's like making multiple copies of the DNA without actually dividing the cell?
Yep. So you end up with many copies of the DNA bundled together.
Okay, that makes sense.
And because of this unusual structure, they have a distinctive banding pattern.
Banding pattern, like stripes?
Yeah, think of it like a barcode, with dark bands representing tightly packed DNA and lighter bands representing less condensed regions.
Okay, so the bands tell us something about how the DNA is packed.
Exactly. But here's the really cool part. When a gene is being transcribed, the corresponding band puffs out.
Puffs out, like it's swelling.
Yep. We call these puffs Balbiani rings.
Ibiani rings, interesting name.
They're a visual indicator of where the DNA has uncoiled to allow the transcriptional machinery to do its thing.
So it's like the chromosome is saying, hey, I'm busy making RNA over here.
Exactly. And by studying these puffs, we can track how gene activity changes over time.
That's fascinating.
It's like watching a cellular orchestra with different genes playing their parts.
So we've gone from simple base pairs to these intricate chromosome structures. It's mind-blowing how much information is packed into something so small.
It really is. And all of this organization allows for the precise control of gene expression, leading to the incredible diversity of life we see around us.
It's like each cell is a tiny universe with its own complex set of rules.
It is. And we're only just beginning to understand all the intricacies.
I can't wait to learn more.
Yeah.
And, it's not just about the structure of the DNA itself.
Oh, really?
Even the environment inside a cell can impact how that DNA, behaves.
What kind of environmental factors?
Well, things like temperature,
Temperature? Okay.
pH, even the salt concentration.
Right.
All of those things can influence how tightly that DNA is coiled. And even whether its two strands stay together.
So you're telling me that, something like temperature could actually cause the DNA strands to separate?
Exactly. It's called DNA denaturation.
Denaturation, that sounds kind of scary.
That sounds dramatic, yeah.
Does that mean the DNA is, damaged?
Not necessarily. Think of it more like unzipping a zipper.
Oh, okay.
Those two strands of that double helix, they come apart because the bonds holding them together get disrupted.
So it's like undoing the spiral staircase, separating the handrails.
Yeah, exactly like that. And heat is a common way to do that.
Heat. Oh,
Think about what happens when you boil an egg.
Yeah, it gets hard.
The heat denatures the proteins.
Oh.
And it can do the same thing to DNA.
So that's why cooked food changes texture.
You got,
You're denaturing all the proteins and DNA.
but that doesn't mean that the genetic information is lost.
Okay, good.
You're just changing the physical structure, not the sequence of bases.
Okay.
And under the right conditions, denatured DNA can actually reanneal.
Reanneal, so put it back together.
Yeah, the strands can zip right back together.
So it's reversible.
Exactly.
Yeah.
But, this denaturing process is actually really valuable for scientists.
Oh, how so?
Well, it helps us study DNA.
Okay.
For example, when those DNA strands separate, they actually absorb more UV light.
Really?
Yeah, and we can measure that.
Interesting.
We can use that to figure out things like the DNA's base composition.
Huh. It's DNA has its own little signal
It does.
that tells us what's going on.
Exactly. And there's a specific temperature at which, half of the DNA is denatured.
Okay.
We call that the melting temperature, or TM.
Melting temperature. Okay.
And the more G and C bases a DNA molecule has,
G and C.
the higher its T.
Why is that?
Well, because G and C base pairs have 3 hydrogen bonds,
Okay.
and A and T only have 2.
Oh, I see.
So it takes more energy, more heat to break those GC pairs.
Makes sense.
But denaturation and renaturation, they're not just things that happen in a lab.
Right.
They happen naturally in cells all the time.
And what's their purpose there?
Well, they're essential for a lot of things cells do, like replicating their DNA,
Oh, okay.
transcribing genes into RNA, even repairing damaged DNA.
So they're kind of like essential tools for the cell to function.
Exactly.
Yeah.
It highlights that dynamic nature of DNA.
Right. It's not just a static blueprint.
It's always changing.
It's like a blueprint that can rewrite itself.
Yeah. And, we've talked about how DNA is packed
That's right.
to fit inside a cell, but inside the nucleus, there's a whole other level of organization going on.
Okay. I'm intrigued.
It's like we zoomed in from base pairs to supercoiling to chromosomes, and now
What's
we're
in it?
zooming out even further. You've got your DNA packaged into those chromosomes,
Right.
but they're not just floating around randomly in the nucleus.
No.
They're attached to a scaffolding system.
Wait, so there's like a physical framework in there that the DNA is anchored to?
Yeah.
I never pictured that.
It's sometimes called the nuclear matrix or nuclear scaffold,
Okay.
and it's a network of proteins and fibers that provides those anchor points for the DNA.
Anchor point, so like it's holding it in place.
Yeah, it helps organize it into these loops and domains.
It's like the DNA is like draped around this cellular scaffolding.
Exactly.
Wow, are these attachment points random or?
Not at all.
Okay.
There are specific DNA sequences that act like tethers,
Okay.
anchoring the DNA to that framework.
So each chromosome has like a designated spot on this scaffold.
They do, and these sequences are called matrix attachment regions or scaffold attachment regions.
Mars and SAR.
Exactly.
Are those like addresses on the DNA?
You can think of it that way. And while they don't have like a strict, easily recognizable sequence like a zip code, they tend to be rich in A and T bases.
Interesting. So what's the point of all this attachment?
Well, think about it. If chromosomes were just floating around freely, they could get tangled up.
Oh, that's true.
They could even break during cell division.
Yeah, that wouldn't be good.
This attachment helps keep things organized, and it protects the DNA.
Okay, so it's for protection and organization.
Exactly. And it also plays a role in controlling gene expression.
Wait, so the location of a gene on the scaffold can affect whether it's turned on or off?
Exactly. Genes that are located near those MARS or SARS tend to be more actively transcribed
Why is that?
because they're more accessible
They're closer to the action?
to the machinery that reads and copies the DNA.
Oh, okay, that makes sense. And what about chromatin and heterochromatin?
Yeah, remember the open stacks versus the archives?
Of the genetic library, right?
Well, the organization of that chromatin into those two forms is also influenced by how it interacts with that nuclear matrix.
Oh, interesting.
For instance, heterochromatin, with its silenced genes, is often found near the nuclear envelope,
Oh, okay.
whereas euchromatin, with its active genes, is more centrally located.
So there's a spatial pattern to gene activity.
There is.
It's like a city with different neighborhoods.
Exactly. And this organization, it's not static.
Right.
It can change in response to all kinds of signals.
Like what?
Cellular signals
Yeah.
or developmental cues.
So it's dynamic.
Very dynamic. It's how cells fine-tune gene expression to meet their needs.
Wow, it's really making me think about the nucleus in a whole new way.
Me too.
It's not just this empty space, it's like a bustling city.
Exactly. And speaking of neighborhoods, we've got to talk about TADs.
TADs, what are those?
Topologically associating domains.
Okay.
They've really revolutionized our understanding of how the genome is organized.
Okay, I'm all ears.
Imagine dividing a chromosome into all these distinct neighborhoods,
Okay.
each with their own set of rules.
Oh, interesting.
That's essentially what TADs are.
So we're going from a citywide view to like a neighborhood level.
Exactly. And what makes these TADs special is that they are regions of DNA that preferentially interact with each other.
So they like to hang out together.
They do,
Okay.
meaning they're more likely to loop and fold together.
Oh, interesting.
They create these little self-contained units of organization.
Little regulatory zones.
Exactly. And they're not just random clusters either.
Okay.
They have boundaries,
Boundaries. Okay.
often marked by proteins called insulators.
Insulator proteins.
Yeah, think of them like fences.
Fences keeping them separated.
Yeah, preventing those interactions between neighboring TADs.
So if Mars and SARS are like addresses, these insulators are like fences.
Perfect analogy. And just like a city's neighborhoods can influence the lives of its residents, TADs play a crucial role in regulating gene expression.
Okay, I'm seeing how this works. So the genes within a TAD, are they like more likely to be turned on or off together?
Exactly.
Yeah.
And it goes even deeper than that. TADs can actually help bring together regulatory elements that control gene expression, even if those elements are really far apart on the DNA strand.
Wait, so you're telling me that something that's far apart on the linear DNA sequence can end up close together in 3D space?
Exactly, because of these TADs.
Because they're folded together?
Exactly. Wow, that's crazy. And why is that important?
Because those elements, they need to be close together to actually interact and regulate gene activity?
So TADs are like matchmakers,
I like that.
bringing together the right regulatory elements to control gene expression.
That's a great way to put it.
It's a very intricate dance happening within these neighborhoods.
It is, and when those TAD boundaries break down,
Breakdown, okay.
like when those fences get knocked over, things can get messy.
Messy, okay.
You can have things like cancer developing because genes start interacting with the wrong neighbors.
Oh, wow.
So you can see how this seemingly abstract concept of these DNA loops and domains can have very real consequences.
Absolutely. Understanding how these TADs function and how they're regulated is a huge area of research these days.
Yeah, it sounds like it.
It has implications for everything from developmental biology to cancer research.
This is really amazing stuff.
It is.
There's so much more to DNA than just that sequence of bases.
You got it.
It's this whole 3D world with neighborhoods and fences and matchmakers.
And all of this organization is so important for making sure that the right genes are expressed at the right time and in the right place.
It really is. But you know what? We've talked about how DNA is structured, how it's organized.
Right.
But what about like the timing of replication?
Oh, that's a good point.
Like, is there a specific schedule for when different parts of the genome are replicated?
There is.
Oh.
It's not just a random free-for-all.
Okay.
We call this replication timing.
Replication timing. Okay. So is there a reason behind the order?
There is a pattern.
Okay.
The early replicating regions tend to be gene-rich and associated with active transcription.
So those are the ones that are copied first.
Exactly. They get copied first.
Makes sense.
And then the later replicating regions are often gene-poor and heterochromatic.
So like the less essential stuff.
Yeah, containing those silence genes that we talked about earlier.
So it's like a priority system.
Yeah, the essential genes get copied first.
Okay.
And the less active ones, they kind of wait their turn.
Makes sense. And this replication timing program, is that like set in stone?
Pretty much. It's established early in development.
Okay.
And it generally stays consistent throughout an organism's life.
So it's a fundamental aspect of how the genome is managed.
Exactly. But if you disrupt this timing, you can have some consequences.
Oh, like what kind of consequences?
Things like developmental defects or even diseases like cancer.
Wow. Okay, so it's really important.
Very important.
But what determines this timing program? Like, how does the cell know which regions to copy first?
That's a great question. And while we don't have all the answers yet, we know that several factors play a role. The structure of the chromatin itself, for example.
Okay, so like euchromatin versus heterochromatin?
Exactly. Euchromatin with that looser packing, it tends to replicate earlier.
Makes sense. It's like those open stacks in the library again.
Right. Easier to access and copy.
Yay.
Compared to that tightly packed heterochromatin.
So location matters too.
It does.
Okay.
Remember those chromosome territories we talked about?
Yeah.
Regions near the center of the nucleus? Those tend to replicate earlier than the ones at the periphery.
So it's like the prime real estate gets copied first.
Exactly. Good analogy.
Cool.
And then you've got specific DNA sequences
All right.
that act as replication origins.
Replication origins? What are those?
Think of them like starting points for the replication machinery.
Oh, okay. So like a designated spot to begin.
Exactly.
Wow. So it's this combination of like chromatin structure, nuclear location, and the specific sequences that determines the whole replication timing program.
It is. It's like a perfectly choreographed dance.
It really is. This whole conversation about replication timing has just, I don't know. It's blown my mind. I never realized there was so much to it.
It's one of those things that seems simple on the surface.
Right.
But the deeper you go, the more you realize just how much intricate regulation is involved.
Absolutely. It's fascinating.
And, speaking of uncovering things,
Yeah.
remember those really cool examples of chromosomes that we talked about earlier?
You mean the lamp brush and polytene chromosome?
Yeah, those are the ones.
Yeah, those are amazing.
We could actually see gene expression happening right in front of us.
It was incredible. Can we talk more about those?
Absolutely.
I'd love to, dive a little deeper into how those structures give us insight into the whole dynamic world of DNA.
Okay, well, let's start with Lamb-Bush chromosomes.
Okay.
Or we find those in developing egg cells.
Right.
They've got that distinctive bottle brush appearance, those loops extending out from the central axis.
Yeah, each loop represents a gene that's actively being transcribed. It's like each gene is waving a little flag saying, hey, look at me.
That's a great way to think about it. And the size and shape of those loops, they can actually tell us a lot about the gene.
Oh, really? Like what?
Well, like how actively it's being transcribed.
So it's not just a static picture.
No, it's a snapshot of gene expression in real time.
That's so cool. And that makes them really valuable for studying developmental processes, right?
Especially in those organisms where they're really prominent, like amphibians.
Right.
It's like we get a window into that whole developmental program as it unfolds.
Cool.
Now, what about those polytene chromosomes?
Those were the giant ones with the banding patterns.
Exactly, found in certain insect tissues.
Right. And they're formed by those multiple rounds of DNA replication without cell division.
So many copies of the DNA all bundled together.
Exactly.
And remember, those bands represent different levels of DNA packing.
The darker ones are more tightly packed. Right?
Probably.
And the lighter bands were less condensed.
And when a gene is actively transcribed, what happens?
The band puffs out.
It does.
It's like, hey, look at me. I'm busy making RNA over here.
You got it. Those puffs, those Belbiani rings, are a visual indicator of those gene expression hotspots.
Hotspots, I like that.
Where the DNA is uncoiled so that all that transcription machinery can access the genes.
So we can actually see where the action is happening.
Exactly. And by studying those puffs, scientists can track how gene activity changes in response to all kinds of signals.
It's amazing.
It really is. It's like watching a cellular orchestra.
All these different instruments playing their parts.
And Lampbrush and polytene chromosomes, those are just two examples of how the structure of DNA and those chromosomes are intimately connected to how they actually work.
This whole deep dive has been such an incredible journey. We've gone from those simple building blocks of DNA to this incredibly complex, dynamic world of supercoiling and scaffolding and chromosome territories, and even time-dependent replication.
It's a lot to take in.
It is, but it's just so fascinating, you know.
It really highlights how DNA is so much more than just a static blueprint.
Absolutely. It's this dynamic, adaptable molecule.
Interacting with its environment in all sorts of ways.
It's orchestrating the complexity of life.
It really is.
It's really humbling to think about.
I agree.
So as we wrap up this deep dive, what's like one key message you want our listener to take away from all of this?
I'd say don't underestimate the power of DNA's structure and organization.
Okay.
Every twist, every turn, every interaction, it all plays a role in the grand scheme of life.
It's a dynamic dance that we're only just beginning to understand.
Exactly.
And as we continue to explore this intricate world, who knows what other discoveries are out there waiting for us?
That's the beauty of science. There's always more to learn, more to uncover.
Absolutely.
And every new discovery, it brings us closer to understanding those fundamental principles that govern life itself.
Beautifully said.
Thank you.
So to our listener, we'll leave you with this final thought.
Okay.
As you go about your day, just remember that within every cell of your body, this incredible dance of DNA is taking place. It's orchestrating the symphony of life.
It is a beautiful thought.
And it's a reminder that even in the smallest of spaces, there exists a world of wonder and complexity waiting to be explored.
Absolutely.